We're going to introduce our next speaker. Many of us know the Smithsonian, but other than artifacts and a wonderful museum, we may not know about the research they do. I had no idea. Our next speaker, Dr. Sara Zapico, is a researcher there, as well as having a day job, as it were, as the assistant professor at the Department of Chemistry and Environmental Sciences at the New Jersey Institute of Technology. Her research interests focus on the application of biochemical techniques to forensic science issues like age at death estimation and the determination of post-mortem interval with implications in aging and biomedical research. Fascinating. It sounds like I'm going to watch an episode of NCIS, Sara. The symposium audience is waiting for more on your topic. Thank you.
Okay, thank you very much for this opportunity and the introduction. Let me share my screen. Can you see it?
We can share your screen. You might want to go to presentation mode in your slideshow menu.
Now, yes, it's fine.
There you go. Yep.
Okay, good. So first of all, yes, I am working in forensic science, but my PhD actually was in biomedical science in cancer research. But I always wanted to be a forensic scientist, so I changed during my postdoc at Smithsonian Institution. Because what I did, and I am doing right now, is translate what I learned during my PhD to forensic science issues. So I am going to go over the different issues we have. There are more, but where we can integrate the omics technologies. So here we have a crime scene that you can watch, for example, on NCIS. And one of the main things will be the identification of the perpetrator from evidence at the crime scene, or sometimes the victim, which is mainly what I do. I am not sure how familiar you are with the identification in forensic science, but we are using what is called in nuclear DNA length polymorphisms. These are repetitions of 4 to 6 nucleotides called short-tandem repeats. And these ones are normally in intros or intragenic spacers between genes. And the number of repetitions in each short-tandem repeats, STR, vary between people. And the combination of this number of repetitions in each STR gives us the DNA profile that I will show you in the next slide. Actually, right now, the FBI has selected 20 core STR loci that must be run in all DNA tests in order to provide a common currency with DNA profiles to upload in the database that maybe you saw on TV as well. So this are not the 20 actually, but this is an example of an STR profile. What you see with the peaks here are the number of repetitions and in the top, the STR. Here, for example, the 21S11, this person is heterozygous with 32 repetitions in one chromosome and in the other one, 38. Here, TH01, you see is only one peak because this person is homozygous, 11. And also, we check the sex through the amelogenic gene. XY means is a male. But what happens in these situations? Because what I show you is normally what you also watch on TV, the perfect profile, but it's not as easy. So many times we have mixtures, moreover in rape cases. Also in mass disaster scenarios, we can have degraded DNA and partial profiles. And it's when genomics, in this case, massive parallel sequence, plays a key role. First of all, I put here the forensic DNA signature prep kit from Verogen because it's the one approved by the FBI to upload in the database. So still, this kit, what it does is to analyze 27 global autosomal STRs, also YSTR, which are from the male chromosome, as you know, but are from the, for example, in cases of in rape cases, etc., and XSTRs from the female. Thanks to the coverage that I show you in the bottom, it's possible to better resolution in challenging samples, for example, the graded samples, and increase the resolution for mixtures, although sometimes it's still difficult. But still, the application of genomics, in this case to forensic science, could help in these challenging situations. But we always need a reference sample, so something to compare. It doesn't matter if we have the profile from the crime scene or the victim if we don't have something to compare, a reference sample from the victim or a reference sample from a possible suspect. In this case, we can think about what is called forensic DNA phenotyping. This means infer the sample donor's external visible characteristics, eye color, hair color, skin color from DNA, from the biological material at the crime scene, or from the unknown bodies. Also, we can include here ancestry and age. In this case, we use SNPs, single nucleotide polymorphisms. As you can see from the picture, are changes in only one base. And the combination of these changes are giving us the different colors, the eye color, the hair color, the skin color, and the ancestry. Here you have comparison between STRs and SNPs. And maybe you think if it will be possible also to identify a person using SNPs only. Yes, and as you can see here, we have the identity SNPs, but you require more SNPs, identity SNPs, to be able to provide an identification. And also, yes, phenotype SNPs and ancestry SNPs. So going back to the to this kit, this has two separate, I would say, kits inside of the kit. So the first one is the one I told you before with the STRs. The second one is this one with the 94 identity SNPs, 22 phenotyping SNPs for hair color, eye color, skin color, and 56 biogeographical ancestry. And here in the picture is the result of the software. But with this, you have to be careful because phenotyping and ancestry analysis are not allowed in some of the states. So moreover, in cases that you are looking for the perpetrator, because SNPs, in this case, phenotyping and biogeographical ancestry, constitutes identifiable information. If you realize when I talk about STRs, STRs are in introns or intragenic spaces. So this one, you don't have, the information doesn't have any meaning, but this one does. So for that reason, in some places, some states, it's really difficult to use this, for example, for identification of the perpetrator, not as much for trying to identify the victim. And the other trend right now that I am sure heard a lot is the lineage SNPs and precisely forensic genetic genealogy. This is based on the looking for identical by descendant SNPs. So the SNPs we share with our ancestors, like here in this family tree. And here you have a table with the relations between the parents, etc. And how does it work? So when you do one of these tests, direct-to-consumer data tests, 23andMe, Ancestry, etc., now you have the option to decide if your data can be stored in the database or not. If this data is stored in the database, GetMatch or family tree DNA, then the forensic genealogists are able to access this data and go try to, for example, from an evidence of the crime scene, to see if you or whoever has a relation with the perpetrator, for example, from the crime scene. And these are the different, I would say, kits or microarrays, depending on the type of kit, depending on the type of kit. You see, they are overlapping. Some of them have more SNPs than others, but at the end, they do almost the same, but looking into different things. And this is the workflow. Basically, when the investigator has a cold case because I am sure you've heard by now, a lot of court cases are solved thanks to forensic genealogy. So they have also to request permission to use the evidence from the crime scene for forensic genealogy. Then, depending on how the things are, there are some labs that they do the whole thing, analyze the SNPs, and then do the genetic genealogy, or others, they simply send to the labs, to analyze the SNPs and send to the genetic genealogy. So they do the microarray for the SNPs or whole genome sequencing, and then they send to the genealogists to do the genetic genealogy analysis. And based on that, then they can follow the trait to see if the suspect could be related to someone in the database. But again, we have another problem here, because forensic genetic genealogy, as forensic DNA phenotyping, provides investigative leads. To correctly identify the victim or the perpetrator, at the end, it requires STR analysis and comparison. So it doesn't matter that you have this in the forensic genetic genealogy. At the end, you need to compare the suspect STR profile with the profile from the crime scene. So again, we have another legal issue here. And I include this one here because we actually use for some cases, mostly degraded and difficult samples, is the analysis of mitochondrial DNA. The only problem it has is inherited from the mother. So all the relatives from the maternal lineage, they have the same mitochondrial DNA. It's not unique because you can, I would say, share, but kind of have the same mutations as other person. And here, what we analyze normally is the control region, which is HV1, HV2, and HV3. So you compare these, the mutations of these regions using normally Sanger sequence with what is called a revised Cambridge reference sequence. And then you compare and note the mutations. With these mutations, you determine the mitochondrial haplogroup, where this, I would say, this person comes from the maternal lineage. So there is here another, this is also from Beroyen. Some labs also are using is the forensic mitochondrial DNA control region and whole genome kit. So they have again two options. You can only analyze control region, but the problem is if the person doesn't have enough mutations in the control region, it's better to get the whole mitochondrial haplogroup to get the whole genome. And this is an example of the results from one of the kits. And also, this is the validation. You can see that they more or less get the same mutations from the control region and the whole genome kit. And we normally use this for this kind of samples that I told you, degraded samples, difficult samples like bone, teeth, hair without root, although now it's possible to get nuclear DNA from hair without root, but before it was more difficult. So normally you get mitochondrial DNA. Fingerprints, for example, because they don't have so much DNA, and burn remains or archaeological samples. So here we cover the genomics and including DNA profile, phenotyping, and genealogy. Going back to our crime scene, the other question is, how long ago the person died? And although on TV maybe used to is easy, actually it's more on one of the most challenging things in forensics. So maybe you are familiar with the terms rigor mortis, livor mortis, algor mortis. These are good, but for determination of post-mortem interval or PMI, but the main problem, they are good for the first days of death, and after that it's more difficult to determine the PMI. And also you can think about insect activity, but also has some disadvantage. And also it's not the same a corpse dying or decomposing here in New Jersey than, for example, in Miami. So one of the things to improve that is what we call thanatobiology. is the use of advances based on DNA, RNA, and protein research for PMI estimation. And this is based on, at the beginning, the cells go to autolysis, but the cell nucleus remains intact for days after death. So we can apply cell biology methods for PMI estimation. And here we are talking first about transcriptomics. And I would like to show you, this is a study from my group. We analyzed the post-mortem interval in rat muscle, and but using actually RNA expression of different genes from autophagy and apoptosis signaling. What we realized was there is a switch in signaling in the first hours of death. At the beginning, there is an autophagy signaling because the cells are going to die, but try to mitigate the stress. But at the end, around 4-6 hours after death, there is the triggering of the apoptosis signaling, triggering the extrinsic and intrinsic pathway, and then releasing oxidative stress and other components that could try to mitigate that. So this was really interesting because of the switching signaling. So when I was actually a student for Pine Biotech, I did, as a final project, I did this small study with previous data from rat retina. And if you are interested in the whole study, is in the YouTube channel, Application of Transcriptomics and Machine Learning to Postmortem Interval Estimation. And here I would like to show you the volcanoplots that we developed. So you can see how, from the beginning of the death, there are kind of changes in the expression of the different genes until the end. 24 hours is up to where they get sampled. You see more genes are expressed. And what I really like, because is related to my previous research, is that when we analyze the pathways affected, here you can see apoptosis pathways, some genes were overexpressed and others downexpressed. So as you can see, this one more or less confirms our previous research. So we talk about transcriptomics, and now the next one, and also one of the trends right now, is proteomics, analysis of protein abundance in postmortem. There are more studies, but this one is more or less recent, and also following with the mouse sapien. You can see how these are different animals, I will say, but they have similar outcomes in terms of proteomics, and also some of the proteins overlap, as you can see in the B. And in fact, they identify 2 proteins that decrease with the post-mortem interval in both animals, JPDH and EEF1A2. So this can be useful for analyzing the post-mortem interval in this case. So yeah, we have the proteome. And then another trend is the metabolomics, analysis of metabolites as a consequence of the decomposition process. And here I would like to show you, this is from 2020, but I think it's really interesting because here they could do the study with human subjects without killing anybody. They did at the Texas State University Forensic Anthropology Research Facility, so body donations, and compare with the rats. And as you can see in the top, you have 2 human subjects. Over time, you can see how there is an increase in these metabolites. Some of them are amino acids, others are decompositional byproducts. And also in rats, you see mostly amino acids, free amino acids. So it could be useful in both rats and human subjects for PMI. So here we saw that proteomics and metabolomics can help for PMI estimation. The other approach for PMI estimation is the analysis of Thanaton microbiome. And this fits very well with the previous talk, because this is related to the analysis of changes in bacterial communities as a consequence of the decomposition process. So analysis here applying metagenomics. And here I would like to show you two studies from my colleague, Dr. Sara C. She did also these studies with human bodies from the Forensic Anthropology Research Facility at the University of Tennessee, Knoxville. And in this first study, she analyzed how the soil microbiome changed with the decomposition of the corpses. And as you can see in the three subjects, in this case called three donors, there you can see the same pattern. At the beginning, there are Proteobacteria, Firmicutes, Bacteroidetes, and others. At the end, mostly is Proteobacteria and Firmicutes in the three of them, with variability, because obviously there is some variability among the subject, but still you see the trend. But also what is really amazing is that in the second study, she analyzed the oral microbiome of the corpses. So see how this changes with the PMI. And you can see more or less the same trend in the three donors, but also you see at the end, the most common taxa or phyla is the Proteobacteria and Firmicutes. So these are really interesting studies, because also we can apply microbial analysis to the determination of the PMI. And in fact, Dr. Asturias and I are going next week to the Forensic Anthropology Research Facility at Western Carolina University to continue with this research and the transcriptomic research. So here we talk about metagenomics for PMI estimation. And going back to our crime scene, and although I said this here, it will be one of the first things you try to do at crime scenes is identification of body fluids. Although you think it could be easy, it's not sometimes, because what it looks like to be blood, it could be ketchup, for example. So in this case, people approach this with proteomics analysis. There are different techniques, mainly MALDI-TOF or Q-TOF, and it's based on the identification of proteins or combination of proteins, characteristics of the body fluid. However, it's difficult to translate to the field, because normally when we go to a crime scene, we try to detect body fluids using alternative light sources, and then when you identify that, you analyze on site, sometimes if not, you send to the lab with, for example, rapid immunochromatographic tests, which are similar to the COVID rapid test that we use right now. So these are similar for blood, semen, saliva, etc. However, this proteomics analysis could be a confirmatory test for body fluid identification. And here you have a summary of the different proteins analyzing in the different main body fluids that you find at the crime scene. But I would like to show you like two examples. This one they did with MALDI-TOF. As you can see, they didn't analyze the whole spectra as previously, but you see the difference in the pattern between seminogenin, which is a protein from semen, versus alpha amylase, which is from saliva. You can see the pattern in each one in the mass spec. In this other study, you can see also the different proteins and the relative intensity or relative abundance. And in this case, the authors also, they analyze, pass through the Q-TOF samples from crime scene. So evidence samples, and even what they analyze, the different proteins, match well with the body fluid. For example, semen, and also semen in different substrates, cotton, leather. So even with that, having the body fluid in a substrate, it doesn't matter, because you can still identify the body fluid. So here we can include in the proteomics, metabolomics, the body fluid identification. So in conclusion, omics technologies can help to solve current issues in forensic science, like the identification of challenging samples and improving post-mortem interval estimation, among others. But omics approaches are slowly integrated into the forensic field due to legal limitations, as I described at the beginning. Future research will be able to expand these current studies, hopefully leading to a wider implementation of omics technologies. And as I told you, from our part, we are going to continue with research in transcriptomics and microbiome. As I told you, with Dr. Sara C. I haven't talked about epigenetics, but I am using epigenetics for age estimation from teeth. But the integration of next generation sequencing technologies in this case is slowly as well. And also is using epigenetics for body fluid identification. Also, another thing I didn't mention, but is also still on the works, microbial forensics for human identification and body fluid identification. So here we need to include these, epigenetics and metagenomics. And I would like to thank first you all for listening, all the people involved in the postmortem interval project, also Bio for the invitation and also for their help during the course I was doing. And one thing I am hiring, we are looking for a postdoc, biologist, biochemist, bioinformatics, someone who knows how to work with this data. It doesn't need to be a bioinformatics, but and also if you are interested in these topics. Thank you. You have any questions, I will be happy to answer now, or later you can send me an email. Thank you.
Thank you, Sara. Forensics is such a captivating field. You know, we see it in TV, like I said, in movies, and yet so much more. Appreciate your time and knowledge for sharing with us. We do have a couple questions for you in our time that we've got left, so feel free to expand. How do forensic sciences benefit from the omics technologies?
So in general, for example, at the beginning, when I talk about genomics, in this case, because of the coverage of the omics technologies, is I would say is easier or more like coverage more in terms of more for example degraded samples. So the problem when we try to amplify the STRs in general is that sometimes the primers are really but are not closer enough to, in the region. So integrated samples is one of the problems that you have. So with the coverage of the omics technologies, in genomics in this case, it's possible to cover this region several times, analyze several times, and try to be more precise and getting better profiles, for example. And then in post-mortem interval estimation, in this case, in general, transcriptomics, proteomics, etc., could help us the different studies described in improving the estimations.
Fascinating. So what are the skills required to establish a career in this field?
In forensic, okay, these are different skills. I mean, depending also on what you. Now, it's funny because I am coming from biomedical science. I mean, I am biologist and biochemist. In Spain, I am originally from Spain. In Spain, we don't have so much in forensics, so I did my PhD in biomedical science. Still, I could change. So I did a master's in forensic anthropology and genetics at the University of Granada, and I changed to do my postdoc to forensic science. So I think the most important thing is you are interested in this field, you are willing to learn, and you have an open mind. There are a lot of legal limitations in forensics, as I described, but I am sure doing research will help with so many things. For example, identification of human remains. So basically, yes, if you are interested in DNA, I mean, biology, biochemistry, yes, it's basically these are the skills.
you're gonna have to know a lot of that. I'm getting from all the speakers that it's just not, you got to just go know this. You've got to know a whole bunch of stuff, right?
Yeah.
Yeah.
Yes.
So
Yes.
I'm gonna come back to real questions. I've got a question of my own. When are we going to see you on TV?
I know.
Okay,
Good
back to
question.
real questions. So what are the current omics technologies used for data from forensic science?
So you mean in real forensic cases? It means because in forensic cases, I know for a fact that some people are using I mean, Verogen is not paying me for saying that, but it's because it's the one approved by the FBI. I know some forensic labs, they use the Verogen kit, the forensic signature kit. Also, others, the STRs. Others use the mitochondrial DNA for sure. And also, because it's one of the things you heard a lot, the forensic genealogy. So applicable to forensic cases. Then in research, we are using all the techniques that I show you today. So one thing is research, but in terms of forensic cases, people are using the omics, the genomics forensic kit, then the forensic genealogy. Mm-hmm.
Okay. How did you get interested in this? What made you switch to this field?
Actually, I wanted to be a forensic scientist since I was a child. But again, the problem was in Spain, there were not so many opportunities. So, yeah, I got my PhD. I mean, I am grateful for the time during my PhD. I learned a lot. And in fact, what you saw today related to the postmortem interval, the RNA expression analysis was because of what I was doing during my PhD. But I really want, I know, and also some people will tell you that, yeah, the funding in forensic is not great, but still I think you are helping people. So I think this is the most important thing for me.
Thank you so much. We really appreciate your time here. I think, at least I think everybody's fascinated because of all the TV shows and movies, but it's still fascinating that all that can be determined in that way. So we really appreciate your time here, Sara. Thank you for being on the symposium, and we'll bring you back here in a few minutes for the panel at the end. So stick around. We'll be right back to you. Thank you.
Thank you so much. Thank you.
You're welcome.